looks good. Okay, so take two. This is streaking out a frozen stock of cells. I'll take a small dab from this liquid, this minus 80 stock we have, and put it onto a sterile toothpick, and then put a dab into a corner of a LB ampicillin petri dish. I'll take a second toothpick that's also sterile and streak that small dab of E. coli across the agar surface. And then I'll take a third toothpick and spread out the very last bit of cells for single colonies, and we'll grow that at 37 degrees overnight. This is a quick demonstration about how to set up liquid overnights. The way I like to do it is to set up one tube that has enough LB and ampicillin in it for all of the cultures you're going to set up. So aliquot, in this case, 12.5 mils to one tube. Add enough ampicillin to that tube for a 1 to 1,000 dilution. Give a quick vortex to mix, or you could pipette up and down. And take a new pipette, an aliquot, an equal amount between the tubes you need. And now in terms of putting in the bacteria, there are a couple of different ways to do it. One is to take a sterile tip, scrape up a colony from the agar, and throw it in. Another way is to take a sterile toothpick, scrape up a colony, throw it in. Another way would be to take a loop and heat it up so that it's red hot. Touch it somewhere to the plate to cool it a smidge. Scrape up some cells and inoculate that way. Clean off the loop. And then you can grow your cultures overnight on a roller wheel. And in the morning, the dense culture will look cloudy, the ones you've inoculated, and if you do a no cells control, it should stay clear. So what I'll do here is try to set up the Oticoli project with 300 milliliters of LB that's been sent or that you have. You would add ampicillin to that. As well as isoamyl alcohol. This can be swirled to mix it. And then 50 milliliters can be poured into a sterile conical tube, or let's say just under 50, let's say 45 milliliters. And into that, we can add one of the strains. This is 1-1 that's been grown overnight. mixed and then added to a flask. This is a 125 milliliter sterile flask. And if you're doing the 1-day version of this experiment, I usually keep 10 milliliters behind as the lag phase control. And then this can be grown at room temperature for about 6 hours to reach log phase, and then overnight to reach stationary phase. And at each stage, you can remove 15 ml to store in the fridge. Those samples stay for about a good 2 weeks. Now I'll show you growing room temperature in a shaker. You can also do it on a stir plate with a stir bar. Okay, we can go. So this is the technique that I use for patching the cells to do the transformation lab. Once they've grown on a petri dish as single colonies, I take a sterile toothpick and pick up a colony and move it to a new LB plate where I scrape the cells in a small square. So I scrape them left to right and turn the plate 90 degrees and scrape them left to right. And they will grow as a small patch like that is big enough, generating enough cells that the students then can use in the transformation lab. After an overnight growth, each of these patches will look something like that. And you can grow these 6 on a plate or so. I would keep the strains 4-1 and 4-2 on separate plates, partially just to keep things from getting cross-contaminated, and also just a little bit easier for the students to find their cells that they need when they run their labs. Okay. So this is a demonstration for how you might run the Colorful World experiment that's on the BioBuilder site. The first thing that you or the students could do would be to mix the solution that's used for making the cells competent to receive DNA. And so that is a mixture of something called TB, which is just a salt solution, with a small amount of DMSO. DMSO shouldn't be put on ice. It can be left at room temperature. But if you mix 50 microliters of DMSO into 450 microliters of the TB that's cold, that's now your transformation solution. And then that 500 microliters can be split between 2 fresh tubes, which I've labeled 4-1 and 4-2. So I'll put 250 microliters into each of those new tubes. And that's where we're going to put the cells to make them competent. To put the cells in, we use a dowel, a sterile dowel, and take one of these patches of cells that have grown overnight. I can show you how to make these patches. So this patch of cells, I'll just scrape with the edge of a dowel like this, and get a scoopful on the end, and put them into the solution that's here just by swirling it around like that. And then this goes into the hazardous waste. And if you have a vortexer, you can vortex just to mix the cells up thoroughly. If you don't have a vortexer, I think this works fine just by flicking. So that's 4-1. Now we'll do 4-2. Again, you just scrape the cells up, and obviously the students would be doing this in teams. This one will swirl. Vortex if you've got it. And now you can take 75 microliters of that transformation solution and add it to the 2 color generating plasmids that we're studying. So 75 microliters into this tube that has a few microliters of the purple generating plasmid, and 75 microliters into the tube that has the green. And again, you can flick it to mix those. We'll do the same with 4-2. These can sit on ice for a few minutes and then can be added to a heat block for 90 seconds exactly at 42 degrees. If you don't have a heat block, you can use a water bath that's heated up to 42. You'll take them out and you'll put them into a rack at room temperature and add a few microliters, 500 microliters of broth to each of the tubes, and then you can plate them onto the Petri dishes that you've labeled for the various transformations. Okay? All right. Okay, so now we've added the LB to the transformations after heat shocking them, and we have no DNA plus the 2 DNAs that went into strain 4-1, and then no DNA plus the 2 DNA programs that went into strain 4-2. So we have a total of 6 Petri dishes. All of them have LB plus ampicillin in the media. You certainly could plate the other half of these transformation mixes onto LB with no drug and get lawns for all of the cells. But what I'd like to do is just show that if you take 250 microliters out of the transformation mix and put it onto your Petri dish, there are a couple of ways you could spread the cells around. One is to take sterile beads like this that you can roll, whoops, spill all over, roll around the surface of the plate like this, and that will spread the liquid over the surface of the plate, and then these can be put into hazardous waste. There we go. Or into a bleach solution to be re-autoclaved or sterilized again. The other way to do it would be to take the spreader, which many students, many places have. So again, you just put 250 microliters on the surface of the plate, and if you have a glass spreader or a plastic spreader that you can sterilize by passing through a flame, and then you can spread the liquid on the surface of the plate like that. And it just needs to be spread around a little bit and then allowed to sink in before you turn them upside down to grow overnight. Okay. So after 24 hours at 37 degrees, this is what a typical transformation series would look like, where the no DNA controls have no colonies growing, where the purple and the green transformations into the strain called 4-1 have a good number of colonies with differences in color that's obvious, and then the purple and the green programs into 4-2, the purple does not transform into 4-2, whereas the green does transform but gives much smaller and darker colonies than the green into 4-1. And so it could be interesting to explore the reasons, what the backgrounds are in 4-1 and 4-2 that give rise to these differences, and try to come up with experiments that could explain what you see.